in a session at SITC where the AV went down for like 10 minutes, and there was a trainee giving the talk, and you just see her sweating up there the whole time. So I really appreciate the opportunity to speak today. This is a topic I've spoken on a few times in the past, and probably many of you actually heard this before, but I think I'm going to talk about anti-PD-1 being combined with IDO inhibitor, but maybe specifically epacadostat and lessons that we've learned in that and how it might inform how we do immuno-oncology drug development in the future. So these are my disclosures. I try to work with all drug companies. So again, many of you have seen this, if not all of you before. Sort of the fundamental rationale for why we think anti-PD-1 works is that some tumors have a T-cell inflamed phenotype where all the activated things that we want to go after to block PD-1 are present. Some tumors do not have that. PD-1 works preferentially on the left-hand side in inflamed tumors. So when we start to think about combinations, it's rational, I think, to say, like, well, what else is in an inflamed tumor as a combination partner that we could try to drug? And the seminal interferon responsive gene, interferon driving inflamed tumors, is indoleamine dioxygenase. And you can see that in the bottom, and this is a figure made by Stephanie Springer and Tom Gajewski some years ago. And that group went on to look at IDO inhibition with a preclinical molecule similar to the drug epacadostat as the first IDO inhibitor in class. And you can see in a preclinical mouse model, PD-1 plus IDO seems to have a combination benefit, and that was a rationale then to bring this kind of strategy forward in clinic. And the initial data for epacadostat with pembrolizumab looked quite impressive, and so this was dating back to 2015 kind of range, and you can see the waterfall plot there, where early in this clinical trial we were seeing up to 60% response rates, and that was in the context of coming in thinking PD-1 was working maybe 35 to 40% of the time. Maybe this seemed like a big deal, and you look on the bottom, you see these are durable responses over time. And I had to take out a lot of slides because of the time, but there are data that look sort of like this in many different tumor types, actually, that look pretty attractive. But of course, all of you know that turned out to not really go so well. So these are the phase 3 data from the ECHO 301 clinical trial, where I think it's actually kind of amazing exactly how all of the curves overlap, just like one for one all the way through, really emphasizing that the drug actually had no potency whatsoever. It was, for all intents and purposes, a placebo drug. The toxicity is exactly the same. The outcomes of the patient is exactly the same. It's sort of like a Pembro versus Pembro trial. And so the question then becomes, why did this happen? And why did all of us let this happen, right? And how do we think about not having this happen again? It's just worth pointing out quickly that IDO inhibition as a class is not just epacadostat, but rather there were a number of in-class agents that were being developed at that time, all of which were quite different. And yet all of them basically got discontinued within about 15 minutes of the press disclosure of ECHO 301. And so why did epacadostat fail? And so obvious questions could be, were the populations different between phase 1, 2, and phase 3? Was there a problem? Was there a lack of monotherapy activity that we could think about? Did they get the right dose going into a phase 3 clinical trial? Was the potency too low? In other words, did we not suppress the target? And that's sort of something I asked about a little bit earlier. Or is just IDO1 a bad target? It actually doesn't have any therapeutic effect. Even if you blocked it as best you can, it still wouldn't make any difference because it doesn't matter. So very quickly, I'm going to speed through some of these because many of you already know these answers, but these were the populations from the phase 2 and the phase 3 data for epacadostat and Pembro. I won't go over the numbers. They're about the same. They aren't that different. Is this lack of monotherapy activity a problem? And so I would argue it is a problem. And so that isn't to say that people are, I don't agree that it could be the case that you could identify a drug that doesn't have a great amount of activity on its own, but you better be able to show that somehow, that it really does something before you take it into a combo. So these were the phase 2 data for monotherapy epacadostat. And on the left, you see the summary of response, big zeros there for any responders. There was some stable disease in a third of patients, and an argument was made when they looked at these, well, when patients were coming off of IO, it looked like maybe they did better, and okay, I guess so, all right? But what about pharmacodynamic studies that actually say, yes, this monotherapy is doing something despite the fact we're not seeing clinical responses? And again, same for the same paper, the phase 1-2 study. These were the data looking at the doses in dose escalation from 50 to 700 milligrams. I have a pointer down here, right? And what you can see is that they can suppress kynurenine as the output marker, but not completely. And so if you think about in oncology, when we think about developing targeted therapies, you need to really get the drug to like IC90s in clinic in order to actually see a therapeutic benefit. And here, it's less than 50% suppression in the peripheral blood, okay? And that's shown on the right-hand side where you see this plateauing effect where you get only about 50% in the peripheral blood. And again, this is not in the tumor, this is in the blood. So suppressing the molecule 50% in the peripheral blood. Now, I guess it's important to point out again, epacadostat is not IDO inhibition in totality, and there were a number of other molecules actually in clinic, and one I worked on a lot was this one, BMS 986205, which became linrodostat, and in that study we actually did do tumor biopsies, and interestingly, if you look, we actually did completely suppress kynurenine in most of the tumors with this drug. But when we looked in the peripheral blood, we actually saw that same plateauing effect, again suggesting that the peripheral blood is not a good place to measure immuno-oncology effects. And so I would really emphasize this as you're thinking about assays you might develop for new drugs. I'm not sure you can use the peripheral blood to actually tell you whether or not the drug is being effective. One thing that was also different about this drug, BMS 986205, was that whereas epacadostat, functionally speaking, had no toxicity profile, there were dose-limiting toxicities with this drug, and we did not pick the highest dose because it was limited by hepatotoxicity and rash, both of which appeared to be immune-mediated. So before epacadostat ECHO 301 failed, Bristol Myers Squibb was excited about these data and didn't want to be second to the field, and so launched a global phase 3 clinical trial of IDO inhibitor from BMS with nivolumab versus nivo alone. You'll note I was the principal investigator of that clinical trial. Unfortunately, about, again, 15 minutes after ECHO 301 was reported out as a press release, this trial was discontinued despite the fact there were already 45 patients that were accrued. Now, am I here to say that this would have worked and this would be a new standard of care? No, not necessarily. Just to say, though, that we probably should do drug development better than looking at other companies' press releases as to whether or not we're starting or stopping phase 3 clinical trials. So I think the most important question, though, about IDO inhibition that really gets back to the preclinical development of do we really know what we're doing is this, is actually the oral hydrocarbon receptor the dominant immunoregulatory node in this pathway? And I really love this picture because I think it emphasizes this point. So the whole field was focused on indolamine dioxygenase and TDO as a master regulator of interferon responses that we could block to enhance PD-1, but it's actually only a minor player in this overall pathway that regulates type 1 interferon responses, where the oral hydrocarbon receptor is a sort of master regulator here, and many of you will know AHR biology probably much better even than I do, but it's responsive to a number of ligands in the microbiota and toxins to regulate Tregs and other responses to malfeasant things that happen in our body. And AHR then can trigger gamma interferon responses, also type 1 interferon responses, and this, in fact, might be a much more important immunoregulatory protein in this process. And IDO, or kynurenine, is actually only a minor ligand, actually, that regulates this drug. So why would that matter? Well, maybe we should actually think about drugging AHR in this pathway instead. And so this is just a fancy nature thing because it looks like lots of AHR all over the place, which is true. But the point I was going to make is that there actually are 2 AHR inhibitors in clinic now, one from Bayer and one from a company called Academies. And I didn't have time to update the thing, but at SITC just a couple days ago, we reported the dose escalation in phase 1 clinical trial of IK175, where there's actually monotherapy treatment responders and immune-related toxicities that are significant. Not a lot, but there are to say that this drug actually does have some effect on its own before we start combining it with a PD-1 antibody. And so I guess I tried to emphasize these points that I don't think we should be rushing forward to just try anything that might modulate the immune system. And I think that unfortunately we've had several failures recently that I think really emphasize this point, and I refer to them here as the disciples of the epacadostat school. And so this question of how are we going to have on-treatment target effects that we believe before we go to phase 3 trials, I think is emphasized here. And both of these clinical trials are in melanoma, where a pegylated IL-2, beta-gamma preferential IL-2 called bempegaldesleukin was combined with nivolumab versus nivo. The phase 2 data in PD-1 naive patients looked promising, and supposedly there were biomarker data, but you couldn't really actually differentiate those biomarker data from nivolumab alone. And in this phase 3 trial, actually the experimental arm of 2 drugs was actually worse than giving just nivolumab alone. And so we sometimes think, well, are we really hurting anybody by doing a PD-1 combo? Because at least they're getting the PD-1. Well, it turns out actually that might not be true. And I think when you go back, it looks pretty clear now that all the sort of agents in class with these initial pegylated IL-2s actually don't really functionally have much activity. And that's taken us back to the drawing board. Similarly, in the PD-1 refractory setting, there was a TLR9 agonist called tilcitolimod. It was combined with CTLA-4 antibody. Initially looked promising in the phase 2 trial. First report of the data, response rate was 50%. Second report, it was 30%. Third report, it was 15%. Global phase 3 trial is negative. And again, single-arm studies, lack of target engagement, none of those things were determined before the drug actually went into a seminal trial. So to conclude my talk, so indoleamine dioxygenase 1 is an interferon-linked enzyme. It's clearly immunosuppressive. There's tons of literature about this, but unclear therapeutic value. Epicadostat had poor drug properties and no translational data that informed the failure of the ECHO 301 clinical trial, and it's clear now that we didn't know enough to actually justify doing that trial. But that IDO inhibitor development really showed pharma Me Tooism, which is something that, as an academic, I get to talk about that. But I understand the pressures of having to go fast, but I think this shows that we're not necessarily doing the patients any good, spending a lot of money doing it. And I guess I'd finally say immuno-oncology is not magic, and that the principles of drug development still apply. And we need vigorous preclinical testing, actual establishment of PK/PD relationships, single agent activity, translational evidence to support that proof of concept, and early randomization in our trials, because we've already seen so many failures in the last 10 years. I mean, it's the purpose of having this meeting, that I think we have to really sort of grapple with the fact that we haven't been doing this over the last period of time. So with that, I'll just say thanks very much, and I always put a plug. We just finished SITC, so we're already gearing up for the checkpoints band at ASCO 2023. Just put it in your mind already, Sunday night at ASCO. So thank you very much.